Aloha, everyone. It's my pleasure today to introduce Amanda Irish. Amanda was born and raised in Alaska, and she's been living in Hawaii for several years now. She really developed an appreciation of plant life when she was growing up wandering the woods in Alaska. She graduated with a bachelor's degree in sustainable and ecological horticulture production from Oregon State University in 2013. She lives today with her husband and 2 children, and they working on turning their 6 acres into a sustainable small farm. And that's part of her motivation for doing her internships today and coming to the TCBES program. And she's going to talk about that in more detail. She actually did 2 internships, so she's going to talk about both of them. And while they seem separate in some ways, they're really linked by her interest in invasive species management. So it's been a pleasure to have the opportunity. I was one of her mentors for one of her internships, and it's been a pleasure to be able to work with Amanda, to feel her energy and her passion. And she actually cries because she cares so much about the plants. She really wants them to be happy, and her tears, I think, help them grow and help many other people grow as well. So I'm really happy to introduce Amanda Irish, and I think she's going to take it away now and start her presentation. Thank you.
All right, thank you, Dr. Ostertag. That was really nice. So I'm going to talk to you today about my 2 internships. The first is with Big Island Invasive Species Committee, and the second is with the Liko Nā Pilina Hybrid Restoration Project. And these are the people that are on my internship committee. As you can see, I had quite a few mentors, so I would like to just start things off by a huge shout out to all of you. You really helped make me a better professional and a better person, so thank you for all of your efforts, both professionally and personally. So as far as my presentation outline, I'm going to start with a little bit about me and my motivation to do both of these internships. I'll then go into a little bit of a background about invasive species in Hawaii, continuing on to my internship with the Big Island Invasive Species Committee, followed up by my internship with the Liko Nā Pilina project, and then I'll wrap it all up in the conclusion. My interest in pursuing a master's degree started at the end of my undergraduate program at Oregon State University. It really started to gain traction after my husband got a postdoc position at the Carey Institute of Ecosystem Studies in New York. Being surrounded by ecological research and immersed in conservation was incredibly inspiring, and I wanted to be a part of that community. I got a little bit closer to that dream after we moved to Hawaii in 2015. I'd already looked up the Master's in Science in Tropical Conservation Biology and Environmental Sciences program prior to moving here, and I knew that it was the right fit for me, but at that point, the timing just wasn't quite right. Later that year, I was hired as an agricultural associate in the research department of a large macadamia nut company in Keau. In some ways, that was like coming home. I was surrounded by trees, working with people who took me under their wing and were incredibly generous with their knowledge. Around that same time, my husband and I bought our first home in the Curtistown area, which was very exciting. In both cases, I started to experience a growing awareness of 2 problems that I really wanted to address. With the MacNut Orchard, the company was interested in increasing production in some of the lower producing fields of the orchards. And I really wanted to contribute to a solution, but I lacked the knowledge required to design a study, perform data analysis, and effectively communicate the results in a way that would identify affordable and feasible ways to increase production in a timely manner. At that same time, my husband and I started to work on taking back the property from a few problematic plant species. The property is 3 acres of former agricultural land and is no longer in production. So what grew back was mostly invasive plant species like albizia, strawberry guava, melastoma, melokia, sleeping grass, and guinea grass. My professional background is in agriculture and horticultural production. So although I could see what the problem was, I didn't have the knowledge or experience to know how to fix it. And it didn't take long before the challenges of managing such a degraded ecosystem became really overwhelming, resulting in more questions than answers. These initial observations and challenges grew into two overall objectives. The first was to gain research study experience, including study design, data collection, analysis, and communicating those results. And then the second was to develop an understanding of invasive species ecology and effective land management strategies here in Hawaii. I got pregnant with my second son and started to think seriously about the Tropical Conservation Biology Environmental Science program at the University of Hawaii, Hilo. And so when my youngest turned 1, I started the program. And this was, in order to gain experience and satisfy both of my objectives, I conducted 2 professional internships well in the program. The first was with the Big Island Invasive Species Committee and learning about data and the metrics involved in capturing the effectiveness of control treatments. And the second was working as a field crew leader with Liko Nā Pilina, which is a hybrid ecosystem restoration project. So invasive species and Hawaii. The global scale introduction of new species into ecosystems due to human assistance, deliberate or not, has increased to the point where there's very few ecosystems in the world that are unaffected. Invasive species are introduced species which have the ability to grow and spread in a new range, can have a wide variety of negative effects on ecosystems, which includes altering the hydrology, productivity, nutrient cycling of invaded areas, species diversity, and community outcomes. Invasive plant species are typically able to adapt to a range of habitats, while native plant species tend to be more niche specific, especially on islands like Hawaii, making it harder for native species to adapt to environmental changes. This is different from non-native and non-invasive species. These are also introduced species, but they do not develop into invasive threats to native ecosystems like plants and other species that are identified as invasive. Due to the extreme geographical isolation of the Hawaiian archipelago, the type and amounts of species that can naturally reach the Hawaiian Islands are limited. The oceanic isolation has allowed the evolution of high numbers of endemic organisms, which are species that are only found in a particular area in the world. The lowland wet forests of Hawaii are endangered due to historical and continued introduction of new species and ecosystem alteration by humans. Because the lowland wet forest has been so heavily altered, we don't really have a clear idea of what native species originally inhabited it. So this proves challenging in efforts to restore the native ecosystem, because traditional or historical ecological restoration maintains the goal of returning a degraded or damaged ecosystem to a previous state in which it was considered healthy. Big Island Invasive Species Committee is a project supported by the University of Hawaii Pacific Cooperative Studies Unit. The committee is made up of nonprofit, private, and government organizations, along with individuals who are interested in protecting the state of Hawaii from harmful invasive species. The goal of BIISC is to protect the native forests, agriculture, and communities on the Big Island from invasive species threats. That is achieved through prevention, detection, and eradication of invasive species throughout the Big Island. BIISC has collected data on the eradication efforts in 2005. The data that I had the pleasure of working with for my internship project included the 8 invasive plant species that BIISC has targeted for eradication. That includes smokebush or butterfly bush, pampas grass, silver-leafed cotoneaster, rubber vine, holly, Nile tulip, photina, persica, and Moluccan raspberry. Data collected from 2011 to 2018 includes invasive plant species, site, acres treated, treatment type, presence and absence of flowering, age as in juvenile or adult, canopy structure, which includes overstory, midstory, upper story, and ecosystem type, and then also latitude and longitude. So the purpose of my professional internship with BIISC was to learn about the effectiveness of invasive species control treatments through data analysis using an invasive weed field data set provided by BIISC. The objective of the internship with BIISC was to determine if the invasive species field data set contained metrics useful in evaluating whether the invasive species control efforts were successful. My role and responsibilities, my role was as a graduate student, gaining data analysis experience. And then my responsibility was to produce a technical report, which was submitted at the conclusion of the internship. And so in order to tackle the data analysis aspect of the internship, I adhered to the steps outlined in the data analysis cycle. The data analysis cycle outlines 6 steps in a data-driven project. The first, understanding the issues, understanding the data set, data preparation, exploratory analysis and modeling, validation, and then finally visualization and presentation. And so I used Excel to organize the data set, and then I used RStudio for the statistical analysis. I went through steps 1 through 4, which is understanding the issues, understanding the data set, data preparation, and exploratory analysis and modeling 3 times. I encountered a few challenges with the data set itself, and then also my level of data analysis comprehension at the time. So after 3 unsuccessful attempts to statistically analyze the data, a 4th and final attempt to identify metrics contained in the data set useful in determining the result of the eradication efforts, the decision was made to try a different approach, right? So the first 3, it didn't work. So we changed it up for the 4th. And the data I chose to, so for that, for the 4th set, I chose to focus on 1 species and 1 site. The site is Kapuka 21. And the targeted invasive species is Cotoneaster penosus, which is a fast-growing shrub. It invades native dominated forests, shade tolerant, resprouts after mutilation, and probably most importantly for this site, the fruit is attractive to birds. Kapuka 21, excuse me. Kapuka 21 is located not too far out of Hilo on Saddle Road, just before the Mauna Kea Scenic Viewpoint. This site is also home to the Kaulana Manu Nature Trail and is a popular place for birders to see some of Hawaii Island's native forest birds. So, and you can see some of the endangered ones as well, which is especially why it's important to control this particular plant at this site. So RStudio was used to create a graph showing the change in both juvenile and mature C. penosus plant counts from 2014, when the data collection and treatment started at this site, until 2019. It should be noted that the data collection at this site is ongoing and continued after the completion of this project. So updated data could affect the graphs, although I don't think so, but it could. So you can see for yourself, 2014, the eradication effort started, but both plant counts kept going up until 2016. The mature plant counts dropped off pretty sharply. And then in a following year, in 2017, the immature plant counts also dropped off. So I was, what got me is, why did the plant counts, both mature and immature, continue after eradication efforts started? And so after having conversations with the people at BIISC and then also doing my own research, it was determined that it was most likely the seed bank, that there was still this Cotoneaster penosus in the seed bank, which is very typical of invasive seeds or invasive plants. And then so through this process, I encountered some challenges, but I also really started to get interested in invasive species management strategies. For example, at this particular site, eradication seems to be working. But then I started to think about what about other sites, and what about other species where eradication doesn't work or isn't an option? And I really, really wanted to know more about what type of other management strategies out there. And in particular, I was interested in the invasive resistance management strategy, which brought me to the Liko Nā Pilina internship. And so in order to learn more about invasive species or invasive resistance, I did the second internship with the Liko Nā Pilina project, which is a hybrid restoration research project located on the east side of the island in one of Hawaii's lowland wet forests. So as mentioned previously in the introduction, I briefly mentioned that the Hawaiian lowland wet forests are extremely endangered. And you can see the map on the left shows the distribution of native ecosystems prior to human contact. And the lowland wet forests in particular are the light green, and you can see they have quite a distribution. But then you look over on the right and you can see their distribution, and this is the current distribution, is really drastically shrunk down. The Keaukaha Military Reservation in Hilo, Hawaii, resides within a highly degraded lowland wet forest on a 1,200-year-old lava flow. This is characterized as a closed canopy wet forest, It is one of the few lowland wet forests still in existence in Hawaii. While the overstory does still contain some native tree species, ohia and llama, it is facing increasing pressures from invasive tree species, which are mainly strawberry guava, bingabing, and melastoma. The increasing onslaught of invasive plant species is negatively affecting native seed regeneration at this site. In 2014, the endangered Hawaiian plant Sertanda navaliensis was found in 3 locations at KMR. Sertanda is the largest and most widespread genus in the Gesneraceae family, which has over 500 species dispersed throughout the Pacific and Southeast Asia. As widespread as the genus is, the endemic species within Sertanda are very narrowly dispersed, often occupying only a single valley, island, or archipelago. There are 60 shrub and small tree species endemic to the Hawaiian Islands, which are usually found in wet forests, occasionally mesic forests. About half of the Hawaiian Sertanda species are classified as extinct, rare, or endangered. As an understory plant species, Sertanda navaliensis is very sensitive to light levels and has specific dappled lighting requirements. By February of 2019, all the wild populations at KMR had unfortunately died, and it is suspected to be due to changes in light levels caused by a combination of loss of ohia canopy due to rod, and then also increasing invasive tree canopy. And the invasive tree canopy provides too much shade. Oh, and rod is rapid ohia death. So restoration in ecosystems. Traditional or historical restoration is the process of actively repairing an ecosystem which has been damaged or degraded to some degree by human activity. Some of the strategies include removing invasive or unwanted plant species and animal species, and then reestablishment of endemic and native species. Novel ecosystems are ecosystems with combination and numbers of species within a biome that have never been seen before. Ecosystems need to have 3 main characteristics to be considered novel. And so an invasive species forest could be considered novel. It has to be self-sustaining. And then a hybrid ecosystem is considered to be a combination of both historical and novel ecosystems. So the adaptability of invasive species is one of the primary threats to native species and conservation efforts. Increasingly, conservation managers are tasked with managing ecosystems in varying stages of degradation. Liko Nā Pilina is a hybrid ecosystem project, restoration project, which was started in 2013, located at the Keaukaha Military Reservation in Hilo, Hawaii. The name of the project, Liko Nā Pilina, loosely translates to growing new partnerships. The goal of the project is to restore and protect the degraded lowland wet forest using a hybrid forest restoration strategy, which allows for both non-native and non-invasive species and native species to create plant communities that can provide invasive resistance, increased carbon sequestration, supports native biodiversity and forest regeneration, is suitable for military training exercises, and also requires very little management. A traditional approach was tested at this site, where the invasive plant species were removed and the native species were left intact. The results of the study showed that even with the removal of the invasive species, the resulting regenerating forest was mostly non-native. It was determined that even though there was native seed presence in the seed rain, the seed bank was flooded with invasive species. Without continuous weeding efforts, invasive plant species would continue to invade and replace native species in lowland wet forests. So it was determined that in order to save and protect Hawaiian lowland wet forests from constant and increasing threat of invasive species, new restoration strategies would need to be evaluated. So from this initial study on the effects of non-native species removal in Hawaiian lowland wet forests, Liko Nā Pilina was born. In April 2018, the DLNR DOFA issued an annual permit, which was renewed again in 2019, allowing for the collection and propagation of Sertanda navaliensis for research and species preservation. And then in August of 2018, an NSF grant was awarded to the research team, which facilitated a project looking at the effect of plant functional traits have on multitrophic interactions. The objective of the arthropod functional diversity project was to evaluate the effect of the plant communities on the microarthropod communities thriving there. So for the study design, they used a mix of 9 native species and 11 non-native, non-invasive species, which were then mixed into 4 treatments. That was moderate complementary, moderate redundant, slow complementary, and slow redundant. And so here you can see this is a map that I got to create this term with GIS, showing the different blocks. So there's 4 blocks, and within each block, there's one of each treatment, and then also a reference block. Reference plot. The purpose of my internship, my professional internship with Liko Nā Pilina, was to develop a deeper understanding of lowland wet forest ecology in East Hawaii, hybrid forest restoration, native and non-native, invasive and invasive plant species, care and propagation of endangered Hawaiian plant Sertandra navaliensis, and the necessary metrics and tasks involved in a research project. My objectives entering into the professional internship included grow my knowledge of invasive, non-native, and non-invasive native plants growing in the lowland wet forest plant, and then also the associated ecology at KMR and in Hawaii. I also want to learn about data collection and strategies for forest restoration research, lead field crew and recurring data collection tasks, and then also to contribute to project goal of outplanting the Sertandra navaliensis. My role and responsibilities, my role was as the field crew leader, and the majority of my responsibilities were the leaf collection, phenology, weeding, plot maintenance, and then Sertandra navaliensis. So one of the first things I learned about the KMR study site is that you just never know what you're going to see. So early on one day going in one of the plots, I saw there was a swarm of bees right in the middle of the path, and it was they couldn't have cared less about me doing it. It just was really interesting. I've never ever seen a swarm before, and so that was really interesting. There's also a gentleman there who is training goats and sheep to eat invasive plants, and so this is an ongoing project, and it is really fun to come out of the field on either a short or a long day and get to see these little adorable babies. They're just too cute, and it's just it's really a lot of fun to see the different projects that are going on and the efforts being made as far as conservation here in Hawaii. So as far as the data collection and field work, we used sheets to keep track of the data for weeding and then also for phenology. For litter collection, the picture on the right, you see where you physically go into the field, and there's 20 traps per plot, and you collect those up. And this is a picture of my youngest brother. He came to visit when I was starting the internship, and I said, hey, why don't you come out in the field? It'll be fun, it'll be awesome. And he's a really good sport, but what you probably can't tell from this picture, it was pouring rain all day, and he is soaked to the bone. But he came out more than once, and so I appreciate that. It was a lot of fun. When it comes to the weeding effort, I mean, it might sound really simple. You're just going to weed the plot, right? But part of that is you have to be able to identify the weeds. And so here's just a random 3 that we can find in the plot: giant fern, alstonia, and autograph tree. On the flip side of that, though, you also need to be able to identify the native species, which is the whole point. For we want native seedling regeneration, and that can get a little tricky because sometimes they look similar to other species. Alohe'e, eventually it starts to distinguish itself. You can see here in the Alohe'e picture, the leaves start to curve a little bit. And then on the right, though, Ohia is a little bit trickier. Especially when you're just learning about how to identify seedlings, they don't always look like their adult parents. And strawberry guava seedlings can look a lot like Ohia, especially to new eyes, right? And so Ohia, though, tends to be on nurse logs like this. The stem is reddish. And so, I mean, after you've looked at it for a little while, you can start to figure it out. But initially, there's really some challenges there. So another one of the responsibilities was maintaining the fence line. There's an electric fence that helps keep the ungulates out. And occasionally trees or limbs can fall on that. The picture on the left shows, it's hard to tell, but this is a huge ohia, which was covered in other plants, and it fell on the fence line. And that was not actually one that we were able to remove ourselves. BIISC came out because they also work at that site. And so they came out and removed this tree for us, which was really, really awesome. On the right, you can see I am removing a tiny little ohia tree that fell onto the fence line. And what you can't see is that this is also another day where I talked my brother into coming out. He's in the picture on the left there. And it was pouring rain all day. And so we were soaked, and but still it was good, and we did it together. It was good teamwork, and it was a good bonding experience. And one thing I learned is ohia is a lot harder. It's a lot harder wood than I thought. We had some tools initially that were incredibly ineffective. And so this was a good learning experience on using the right tool for the job. So the Sertandra, probably for me, the most important responsibility I had besides crew safety in the field is taking care of Sertandra. I was initially trained, and then the person who had been training or who had been taking care of it went on vacation, and I was excited. I'm like, okay, I'm taking over. This is great. You know, I'm really getting to get in there. But right off the bat, I came into work. We got a call that there had been a flooding event. And this is, you know, the backstory here, the Sertandra, they started propagating it last year, and there's been some struggles. It's been a real kind of the mortality rate has continued to rise over the last year, but there's been a lot of knowledge gained in this process also. So we got the call, there's been a flooding event. I go in, and you can, every tank is completely flooded. And so that's really disheartening. They had just gone through a heat event where that caused a lot of mortality. And so they had kind of just started to recover. This is a little disheartening, but the picture to really pay attention to is the one on the far right. This was the seedling at the time that had the most promise. It was really looking good. And there was a lot of hope that this plant would continue to survive and thrive. So this was especially devastating to see it floating at the bottom of the tank for who knows how long. So, you know, cleaned it up, got everything repotted, got them in their position. And due to, you know, the knowledge that's already been accumulated about their lighting requirements, thanks to one of my cohort members, James Melcher, and then also the previous experience from my mentor, Nicole Domano, I thought, well, if they like dappled lighting, we have some plants here. I'm going to go ahead and try and create a dappled lighting effect on top of the shade cloth that's already there. Why not? We had the plants. And so that's what I did. And I was, the whole outside layer is hala. And if you're not familiar with hala, it has tiny little, not thorns, but it definitely grabs you, and it teaches you to be very respectful of it. So this is the Sertandra after it was repotted. And what's important about this picture is to pay attention to on the left, the two largest seedlings. They distinguish themselves fairly early on as doing better than all the other ones. And so I had some real hope for these two. And then this is the seedling on the right that was floating in that tank all by itself. It survived, and it didn't seem to suffer too much. And so potted it up, and then this is like a month later. Those two promising little seedlings started to look not so promising. They started to yellow. They clearly weren't doing really well. All of the other little tiny seedlings were dying off. The leaf cuttings were dying off. The stem cuttings had died off. And so the numbers had shrunk considerably. However, the one seedling continued to do really well and show promise. So we got the opportunity to go visit Jamie Anoka at the Rare Plant Facility in Volcano. And she works with a multiple species of Sertandras, and including the Sertandra navaliensis. And so she was kind enough to share with us her propagation techniques, the types of fertilizer she's using, her setup, all of that. And so I took some of that back with me and conferred with both Becky and Nicole about what we should, you know, what should we try and do. And they let me propagate. They let me go ahead and propagate. And it was kind of a risk because at this point, the numbers are down. We don't have the right size leaves, but I felt like there was two plants that could sacrifice one leaf. And if the leaf cuttings look really small, it's because they are. This is a 4-inch pot. Each cutting there is probably a 1/2 inch at most. And that's not ideal, but I just felt like we had to do something. And I felt like this, you know, this could be it. I want to try. This is what I do. This is what I've said I wanted to do. And they let me do it. So, you know, fast forward to COVID and things got a little strange. So fortunately, one of our team members was able to take them home and she took good care of them. Later that week, I was able to get them for her and then brought them to my home where they have continued to do well. And so this is that one seedling that from the very beginning, people had some really high hope for. People, you know, Trevor took really care of it. He's a Pilina member, and Amy also. Really, they all poured some love into this plant, and it has continued to do well and thrive. And it's hard to tell the size difference in this, but I can tell you that this pot is a 4x4. In the previous picture, it was in the little 1-inch cell. And so it has continued to grow, continued to thrive. The picture in the middle is showing this, what's remains of the little seedlings that continue to not do well. And I don't have real high hopes for these ones, but I'm still, I mean, I'm doing my best. I am hoping, but you know. On the right though, this is the picture of those two little seedlings that showed promise and have continued to do well, have continued to thrive. And now they're in their own 1-inch by 1-inch, and they just continue to do well, which is really exciting for me. Most excitingly, I propagated these leaves on February 20th. On April 20th, I saw this. Well, this is a more advanced growth, but I saw that they did indeed have babies growing out of them. Two out of the 5 leaves did. One died off, another one died off. And then so there's 3 left. 2 are sprouting babies. The third, well, no babies, is rooted, and I do expect that it will do that. And so this was probably the most important accomplishment for me, and the biggest way I felt like I could give back to the project. I came into the program saying I want to work with native plants, I want to propagate native plants, and I got the opportunity to not only propagate native plants, but an endangered plant. So for me, professionally and personally, this was really big. It was important, and Becky's right. I get emotional about it because I've been given so much from this place and from this program, and people have been so generous with their knowledge. It's important for me to give back and to show my children that we give back. And so this is just, it's amazing for me. And so, but it doesn't stop here. Right? We've gotten to this point before in the propagation efforts. And so now the goal is to not just propagate, but to continue to help them thrive and grow. And what do they need? What are the conditions? Because not a whole lot is known about this plant. You know, they figured out a few things so far, but there's a lot that we still don't know. So that is the next step. It will be to introduce some fertilizers based on her program up there at the Rare Plant Facility. And so hopefully we can continue to not just propagate, but get these plants to thrive, and then, you know, really accomplish the final goal, which is to outplant them at KMR and return them to where they really ultimately belong. So in conclusion, I love this picture because if you look at the pinky on the right, you see that it's swollen. There's some dirt jammed up into the nail. There's a little scratch there. And this was the result of me tripping. I lost my footing on one of the plots. And I should have said, these plots are very physically challenging. The lava is sharp, and it's sometimes just covered in invasive plant, the leaves. And so you can't really see where you're going. It's hot, it's humid, all of these elements. And one day I just wasn't really paying attention. I was busy talking. I lost my footing a little bit, and I used that pinky to break my fall. And it's still to this day, that's the best that it can do. But it's not about the pinky. It was not about that, because wounds heal. But really what it was about was not losing perspective in the moment. It was about respecting the site, remembering where I was at, and keeping that mental sharpness and focus, because I was there to do a job. And when you're in a plot like this, in a site like this, where there's some elements that you really have to overcome, the physical challenge of the terrain, the, the heat, the different tasks that I'm trying to learn while also, uphold the standard of the project. And so this was my favorite and best reminder to keep that mental sharpness and keep that focus, and drink your water, pay attention to where you're walking, talk less if you have to, which if you know me well, that's probably a challenge. But this site really, in this one moment, and a few others, there's been a few other shin bangs and whatnot, but really taught me to respect the site and what I was doing there. And so for that, I'm incredibly grateful. So in conclusion, I started this presentation off talking about how we have this property, my husband and I, and there's an incredible amount of invasive species here. And some of those are albesias. And albesias are tree species that are incredibly invasive here in Hawaii. They're very fast growing. They change the nutrients in the soil, so they make nitrogen a lot more available, which facilitates more invasive plant species, which use nitrogen a lot more than the native species, so they can outgrow them and outcompete them, and ultimately it results in no native seedling regeneration. So these are the two huge albesia in the back of our property. And working for BIISC, they have an albesia assassin program. And so through conversations with them and their facilitation and training, my family were really motivated to do something about these trees, to stop their spread within our area and just, do our part in our community. So we did. We had a family conservation day. I got to bring my sons, and my husband was the one taking the picture. And so I did most of the work, but I let my 5-year-old do a little bit of chopping with me. And he didn't get to hold the hatchet ever by himself. It was always he and I did it. My baby on the other side, who is 2, didn't get to participate at all. And that made him a little bit sad. But at this point, it's, we're not gonna let a 2-year-old hold a hatchet. So coming full circle, what this all really means to me, the program, the 2 internships, it just really comes down to that it's actually not about me. People have been very generous in sharing their knowledge and their time, and I learned so much. There's been so many lessons. I can't even unpack them in one moment. That'll take years, but it just, for me, boils down to that it's not about me, it's about the keiki, it's about the children, and it's about passing this knowledge on, and not just passing it on verbally, but physically getting in there and doing the work and showing them how to do that too. And so for all of those opportunities, I'm incredibly grateful. And I really appreciate all the time that people took to talk to me, to show me things so that I could then turn around and do this kind of stuff with my family. There's a lot of people who help facilitate not only this project, but also my professional development. I'm not going to read them all name by name, but here they are. I also need to thank my family, and then most importantly, my husband, Tobias. So thank you, mahalo. If you have any questions, there's my email on the bottom, or please, feel free to ask right now.
All right, Amanda, thank you very much for your presentation. That was wonderful. I'm really touched by how much you care about the plants and the environment. So I have a question for you, and it has to do with it's similar to the one that I asked Alex. Where do you see yourself in the far future, and where are you going in the near term?
It's a great question. I have no idea. What I do know is that I love what I do. I really love what I do. I love what I've gotten to do with Liko Nā Pilina, and then the influences also from BIISC, and that's where I want to go and continue. I don't know that it will be with either one of those, but it's definitely going to be in conservation. And then I hope to continue my work with the Sertandra, or if not with the Sertandra, then another native species or endangered species. It's incredibly gratifying and fulfilling, and I feel really good about it. So exactly what avenue that's going to take, I'm not entirely sure, but I will absolutely continue to do this.
Great. And then we have a question for you from Dr. Cordell. She asks, she states, it's been great working with you, Amanda, and she's wondering what your thoughts are about the hybrid approach. Think it's a successful strategy for lowland wet forests?
So I first heard about this project when we started the program, so 2 years ago, and right away I felt like, yes, okay, this is the hybrid approach is a strategy that can be superimposed in other ecosystem types, not just the Hawaiian lowland wet forest, but for example, here on the property, that, you know, taking a hybrid approach could be really effective. And so that's in part why I really like the approach this project took. And I you know, when you're in the plots, it's hard to tell. And so as I become more familiar with the data, it's really enlightening, and I hope it works. It seems like it works. I see areas within the plots that need less weeding than others. And I really hope and look forward to learning more about the specific outplant species and their effect on the invasive resistance. And so thank you, Dr. Kolda. Dr. Kolda, I really appreciate your support and getting to work with you all on this project. It has been amazing. I hope that answered the question.
Yeah, absolutely. Well, thank you very much. Before we segue to the next presentation, do you have any last words of advice for future internship track students or students in general?
No. I ended up doing 2 internships, and there was moments where it felt like I wasn't going to get through it, and it was a lot of work, but the changes that you know, that I have felt professionally and personally have made it worth it. And I would do it again, every part of it. So, and the program, yeah, I'm incredibly grateful for all those opportunities. So thank you. That's really it. Thank you, everyone.
Okay. Well,